PharmaVentures, experts in deals and alliances.
Hello, welcome to Pharma Television News Review here at the International Biofest in Melbourne in Australia in 2016. On this show, I have Michelle Fraser, who is the CEO and Managing Director of a company called Reproductive Health Sciences based in Adelaide here in Australia. Welcome.
Thank you.
We were just discussing before we started how you describe your company, and we sort of said probably a research tools company. So could you tell us a bit more, Michelle, about the company and what your purpose is?
So we originally started out with a product for assessing IVF embryos, and these products are sold under the research use only label, so they're not regulatory approved diagnostics, they're a screen for embryos. The second product that we released is a platform technology for testing single cells, the genetic contents of single cells. So it's a way to amplify up the DNA, and then you can screen it using a range of different downstream methods. So it's a molecular tool that's a platform technology for broad application.
Right. So let's just go to the first product that you've got, and it's, as you say, it's product service. So what did you identify as the unmet clinical need there? Because obviously IVF is a very important part of reproduction for people who have difficulties in conceiving. Mm-hmm. There are problems with that during that process, and it's painful and long term sometimes. So where does your product come in terms of intervention to make sure that actually increases the chances of success?
So IVF embryos are grown in culture up to between 3 and 5 days post-fertilisation. During that time frame, there's an opportunity to biopsy the embryo and then test the genetic contents of that biopsied cell or small number of cells. So you've got somewhere between 1 cell and up to 8 to 10 cells that you can biopsy at various stages of embryo development. When that biopsy is tested, what our first product looks for is, does it have the correct number of chromosomes in that biopsy or not? If it does, then it's suitable for IVF transfer, because you've shown that it's chromosomally healthy.
Correct.
If it's not, it has the incorrect number of chromosomes, too few or too many. 96% of those embryos will not implant, so you have a failed IVF transfer. The 4% that do implant, most of them miscarry, and very few of them will go on to a prenatal screen where there'll be a diagnosis of something like Down syndrome, which is one of the very few viable incorrect number of chromosomes that can happen in an IVF embryo. So what the technology is aimed at doing is trying to focus clinical attention on the embryos that have the best chance of implantation success and live birth outcomes to reduce the number of failed cycles.
So what is the underlying technology that you're using to detect these abnormalities, we call them those?
So the kit involves a lysis and PCR based method to copy up the DNA from the small number of cells, because there's not enough DNA in one cell to be able to do anything with. Then there's a labelling step where you label your test sample with a green fluorescent dye and a reference sample with a red fluorescent dye. They're mixed, and then they compete to hybridise to a glass slide that has chromosomes printed on it. So what you're looking for is there the same amount of green dye bound as red dye? And if there's not, there's an imbalance with the test sample. It's either too green, so there's too many chromosomes, or too red, so there's not enough chromosomes. So it's a very straightforward black and white answer. Does the embryo have the right number of chromosomes or not? So we manufacture the microarray, and it's a patented technology. The approach that we use has been granted in patent in the US, UK, China, Australia, and a few other key territories. And the front end of it, which is the copying part of the kit, is developed by RHS in-house, and it's based on know-how, and we've got exclusive access to the reagents for that part. So it's a fully owned RHS product.
Right, and obviously that's sold worldwide.
Yep.
And how is that doing? How is that product doing in terms of sales and so forth?
So the market has started to move towards next generation sequencing to try and reduce the costs. Microarrays have been in favour for a long time and are slowly falling out of favour, which is one of the reasons why we've done some proof of principle and some validation of our next generation sequencing approach, which leads us into our second product. So the first product has been fully validated. We've released the results for it, but the market has been moving very rapidly towards next generation sequencing.
So your next generation products will be what?
So it's the copying part of the first kit. So it's the lysis and whole genome amplification from single cells or small numbers of cells. Then there's a standard library preparation kit that you can buy from a whole range of different manufacturers, and then you use a sequencer from any of the sequencing companies, Illumina or Thermo Fisher. It's platform agnostic, so what we sell is the very first step, which is the critical step as to whether you get a good result from your sample or not.
Okay, excellent. So, I mean, so from what I understand, you mean you set out as a reproductive health sciences company, but your technology is your ability to look at individual cells and then detect what's inside those cells.
Correct.
Particularly from a chromosomal point of view or genetic point of view.
Yep.
So you diversified out of that, obviously.
Yes.
And so tell us more about how you've diversified and how you see your company today in terms of offering and its overall strategy in terms of where it's going.
So we started out, as you said, with a specific purpose of trying to improve IVF success rates. We've diversified to releasing a platform technology. So the product for lysing and whole genome amplifying single cells was launched in late August this year. And what that opens up is things like looking at circulating tumour cells, circulating fetal cells, looking at tumour diversity by taking multiple biopsies of small numbers of cells or even single cells, and looking at whether there's a genetic difference across the tumour. It allows us basically to sell a product to anyone who's working in the single cell analysis space. So there are only 8 kits on the market that can do single cell whole genome amplification, and we're the only one that use our particular approach. There are others out there, but they don't do it the same way as ours does, which gives us benefits, technical benefits in the market. And we've also been able to combine in there the ability to target specific genes or regions of clinical significance while you're doing that generic copying.
Right.
So we've got another unique advantage over the other kits in the market.
So what's your target now? I mean, obviously, circulating cells in those various settings, all different clinical settings.
Yep.
So what's your coverage there? What settings are you trying to cover for the application of your product here?
So the good thing about being a platform is that we don't have to modify the product for any of those downstream applications, which means we can have multiple parallel conversations going on across a range of different outcomes and different applications. So we're talking to companies that are isolating individual cells, whether they're tumour cells or fetal cells. They don't have the next step of that workflow in-house. They're testing a whole range of different products on the market. So initially it is targeting the circulating tumour cells and fetal cells market, and then we'll expand out from there. But we have groups that are talking with us and collaborating with us at the moment, looking at the ability to pick up genes that are inserted into other cells, like a mouse or a sheep embryo model. As an example, they've inserted Alzheimer's genes, human Alzheimer's genes into them to create a model to then be able to test pharmaceuticals.
Right, okay.
So it's much more open. We don't have to necessarily have our blinkers on and chase just the IVF applications at this point.
So it just opens up this whole area, which is obviously blooming at the moment, because looking at individual cells circulating within the body in various contexts, clinical contexts even, is becoming increasingly important.
Yeah.
So now, how do you describe the company? You still describe it as a research tools company?
Yeah.
And the future is?
So we see ourselves as an enabling company. What we're doing now is assisting other groups to do the proof of principle and the application of the technology, in the hope that they will then go on to talk to other people about it and get that word of mouth marketing. As far as the company positioning going forward, for us to develop new technology, we've been bringing in new technology rather than developing in-house. We also have a service component of the business, which is advanced genetic testing, and whether that's of human samples or not is quite irrelevant from our perspective. We have a very good depth of expertise and understanding in how you handle single cells and small numbers of cells and limited templates. So we're using that expertise now to offer that out to a market who wants to embrace that single cell genetic analysis application, but don't have the ability to do it themselves yet.
Sounds exciting.
So started with the IVF industry and is now growing beyond that.
Michelle Fraser, thank you very much indeed for coming on the show.
Thank you.
PharmaVentures, experts in deals and alliances.